Okay, thank you, Mando, for the introduction, and of course, thanks Animal Behavior Live for this invitation, and thanks to all people that is connected on the YouTube channel. In this talk, I will show you some works that study the division of labor in stingless bees, but from different point of view. Not all the studies are mine, but they talk about behavior, physiology, or morphology. But in my case, my experiments are related with the chemosensory reception. To start, we need to talk about sociality. I'm pretty sure that if I ask you for some examples of social insects, you will say termites, ants, maybe some species of wasp, and of course bees. But what is the definition of eusociality? And there are three main characteristics. For example, one of them is that there is a cooperative care of the brood, there is an overlapping of generation of adults within the colony, and of course, there is a division of labor into reproductive and non-reproductive groups of individuals. Again, maybe the first example that comes to our minds is Apis mellifera, where they have many workers, female workers, one queen, and some males or drones that are present during some seasons. But it's worth to mention that there are more than 20,000 species of bees, and most of them are solitary. But here we are talking about stingless bees, and they are social too. Why stingless bees? Because, I mean, first of all, they don't sting, so for me it's very nice, and also they are cute, but this is my personal point of view. Stingless bees also are very diverse. Here I put these two trees just to show you how diverse they are. This is from a review that I think that was published 2 or 3 weeks ago from Engel and co-workers. What we can see that is that stingless are along the tropical and subtropical areas all around the world. And in this map, you can see the distribution in blue. And I took this map from the book about stingless bees written by Christoph Rutter. And I recommend that if you want to know about the stingless bees, you must read this book. It's amazing. It's almost about everything in stingless bees, and it's just the beginning. But again, stingless bees also have a significant cultural and economic importance. Cultural because as they don't sting, people can have the colonies at home and they can manage the colonies, they can take the honey on the propolis, and of course, in another level, these products have an importance in economics because you can sell honey, and of course, these bees are good pollinators in our systems. So, from now to the end of the talk, I will show you results about the Tetragonisca angustula, that is the species that I study. It is a very small eusocial bee. It's half of a centimeter. The distribution is from Mexico to central Argentina. So I live in Argentina and I do my research here. So it's nice to study native species. And as I told you before, they have an important role as a pollinator of tropical flora and agrosystems. And the other thing that these species has is that if you stay for 1 hour or less in front of the nest, in the entrance nest, you could see that there are bees that are performing different tasks. For example, you can see that there are some foragers. That means that you can see some bees that are carrying pollen on their legs or resins, or maybe they are bringing nectar to the colony. And if you stay watching the entrance, you can see some bees leaving the nest with some pellets of waste on their mouths, and the waste removers. They are cleaning the colony, and of course you have guards that are flying around the entrance, or they are just standing on the entrance tube. But I will show you some videos to explain better this kind of division of labor that you can recognize. I have a video here. I recorded this summer, Argentinian summer in Misiones. It's in the north of Argentina. And what we have here is a tree, and this is the entrance tube of one of the nests. It's made by wax, and it's the only thing that you can see of the nest. This nest is inside of the tree. Here we have some bees that are standing on the tube. These are the standing guards. And here, I promise that you can see it. There are some bees that are hovering around, like floating in the air. They are the hovering guards. And also you can notice that there are some bees entering and leaving the nest. I will play the video. I don't know if you can see, there is like little cloud here. They are the hovering guards, and again, the standing guards. I will show you again, just in case the connection is not good. Again, and we can see also the bees entering and leaving the hive. But if you zoom in the entrance tube, I will show you a video from a colleague. What I want to show you here is that you could see some bees leaving the nest with the waste pellet in the mouth, and also there are some bees entering with pollen in the hind legs. There is one. Here there is another removing the waste. One with pollen. Let me see. And there is one more with pollen. Again, just to be sure that you can see it, I will play it again. The first bees that you see leaving the nest are the waste removers, and then there are 2 or 3 that are entering pollen to the nest. Okay. So here, I mean, I told you that there are some behavioral stuff that you can say, okay, this is a forager of pollen, this is a forager of nectar, or this is a guard. But there are studies that shows some different characteristics that defines the division of flavor in Tetragonisca. For example, a beautiful work from Hamel and co-workers show that there is a temporal polyetism. What means this is that the age can predict which task a worker will perform. For example, young workers perform tasks within the colony. They take care of the queen, or they take care of the brood, they build cells, and the older ones go outside of the nest to defend or to seek for resources. And what we have here is different tasks that bees perform. I will show you some examples. For example, building nests is something that happens during the first days of life. Entering and leaving the nest is something that happens during the end of the life. And for example, guarding is also something that happens at the end of their life. But there is also another trait that is related with the division of labor in this species, and is that as happens in ants, they present minors and majors workers. I mean, this is in terms of body size. Grutter and co-workers, also in an amazing work, they found that there were some minors bees that were the foragers, and the majors were related with the guarding behaviors. Probably they say that this difference in morphology is related to the fact that a larger body could improve the fighting ability. So related with this, also what they show in this graph is in light green are the tasks that the minor workers perform, and in dark green what the major workers perform. And again, for example, guarding is something related with the larger workers. But there is more. It's not only about age or only about the body size. I told you that there are 2 types of guards that you can distinguish because of the behavior. The standing guards were those guards that were on the nest, on the tube entrance to the nest. What some studies show is that they can discriminate against nestmate to non-nestmate using the olfactory cues. While the hovering guards, that are those that are floating or hovering around the nest entrance, they can detect the non-maced using visual cues. So in a recent paper from Valadares and co-workers, they study the brain size of the Tetragonisca angustula, they study the minors, workers that will be foragers, and the two types of guards, the standing and the hovering. They made this beautiful map, and what we have here are the antennal lobes that are related with the olfactory reception of the bees, and related with the antenna, of course, the optic lobes related with the visual cues, and the mushroom bodies that are related with learning. And they made very beautiful descriptions of different areas of the brain, but I want to show you here is that what they found is that the hovering guards present larger brain compartments related with the visual processing, in particular, the medulla. So we have here something that we can distinguish the standing guards because of their behavior, but also there's a correlation in terms of what happens with the brain morphology. So until now, what I told you about the Tetragonisca and division of flavor is that there is a temporal polyetism, that there are some difference in terms of behavior, and there are also difference in terms of morphology, body size, or brain size. But on the other hand, there are some things that we know from Apis mellifera, and that is one of the bees, I think that maybe the most studied bee. We know that in Apis, the division of labor is related with age. There is a temporal polyetism, and that these workers also differ in the response thresholds to different stimuli. And in particular, when you study foragers, there is a correlation between the behavior of foraging for pollen or forage for nectar that correlate with change in the sucrose response threshold of these individuals. That means that, they found that pollen forager present a lower sucrose response threshold, while the nectar foragers present a higher threshold. That means that they need nectar with more concentrated in sucrose to respond than the pollen ones. So with all this in mind, the goal of our experiment was to compare the gustatory and olfactory responses of different workers of Tetragonisca angustula. Foragers and guards, for example. And this experiment was done in the social lab, insect social lab at the University of Buenos Aires. There is a protocol that is very well studied in Apis and also in some ants and even in stingless bees that can give us an idea of how responsive is a bee when we offer different kind of sugar, or actually different concentration of sugars. This protocol uses something that is a reflex response of the bees. That is, if you touch the antenna with sucrose or sugar, they extend the proboscis. I have here a video to show you. Okay, this is a Tetragonisca in a pipette tip. It only can move the antenna and the proboscis. And what we have here is a toothpick with sugar. And the idea is that you see that when we touch the antenna, the bee is going to extend the proboscis. I will play it. So we have it. I will play it again. We touch the antenna, and it extends the proboscis. So with this response, what we can do is to offer different solutions, sucrose solutions, and see to which of them the bee response. For example, we offer 1, 3, 10, 30, and 50% of sucrose solution. And I will give you an example to explain better what I mean. For example, you touch the antenna with sucrose solution of 1% and the bee doesn't extend the proboscis, then you try with 3% and it extends, and then 10, yes, you see the extension of the proboscis, 30 and 50, you see that bee extends the proboscis. So with this response, you can get a score that is related with the response threshold of the bee. For example, here we have a score of 4 because the bee respond or extend the proboscis to 4 of the solutions that I offer. For example, if you try with a different bee and you offer the 1% solution and it doesn't extend the proboscis, then you offer 3 and doesn't extend the proboscis, nothing with 10, nothing with 30, but when you offer 50% of sucrose, the bee extends the proboscis. So the score for this bee is 1. That means that this bee needs the highest sucrose concentration to start responding to the sucrose. So it's important to know here that if you have a high score, means that the threshold is low, and if you have a low score, the threshold is high, because you need the solution with the 50% of sucrose. Okay, so what we did was to compare the gustatory responsiveness of foragers, nectar, pollen, and resin. Something that I haven't told you is that for stingless resins is a very, very important resource because they use it for nest defense and also in nest construction. So it's something that you, again, if you are in front of the nest, you can see bees bringing resins. Also, we test the waste removers and the 2 types of bars. So what we have here is the gustatory score. As I present only 5 concentrations of sugar, the maximum score is 5, and so it's not more than 5, is what I mean. And here we have the foragers, nectar, pollen, and resins, the waste removers, and the guards, hovering and standing guards. And the first thing that we see here is that the nectar foragers present the lowest score. It means that they have a high threshold of response. That means that they were responding in average to the solution of 30 and 50%, where the other groups present a score of between, in average 3 or 5 or 4 or 5. What we have here is something that is the same that happens in Apis mellifera, that nectar foragers have a low score and pollen foragers have a high score because the threshold is lower. But also here, for me, the interesting part is that I told you before that there is a morphological trait that is related with division of labor between minors and guards. And here we have something that even if you are looking at the minors, for example, the foragers, you have differences in thresholds. And these thresholds are more similar to the major bees. So what we can see from here is that nectar foragers present the lowest score. It means that the highest threshold, that pollen foragers and guard present a similar gustatory response score. The waste removers bees are different from pollen. And here we have the resins, the foragers that collect resin, but the sample size is low, so we need more to see if we have some significant difference between groups. But here is another example that the response threshold are different according to tasks. Okay, The next step was to study, okay, we have some differences in terms of gustatory responsiveness, but now we want to see what happens with the olfactory responses of foragers and guards. In this case, we only study nectar and pollen foragers and standing and hovering guards. To do this, we use electroantennography. That is a very nice methodology that allows us to study the detection of volatiles that the antenna can perceive. And I will show you very quickly what means this. You can cut the antenna of the bee and put it in between two electrodes, and one on the tip of the antenna and one on the base. And these tips are connected to amplifier and to a computer, and you offer a stream of air. And what you can see is when you offer the air, what happens with the response of the antenna. For example, this is a response, probably is a mechanical response, but if you add some odors to the air stream, what you get is this kind of response. What you record here is that the small voltage fluctuations between the tip and the base of the antenna, and these fluctuations happen during the stimulation with air or odors. These fluctuations of the voltage are related with the polarization of the olfactory neurons in the antenna, and of course the amplitude of the response is dependent on the nature of the stimuli, for example, concentration of odor. So what we did was to use two different odors. We choose non-anal and citral. They are two very common odors in floral bouquets, but there is something in particular related to the citral, is that citral triggers a defensive behavior in Tetragonisca angustula because citral is present in the mandibular gland of a robber bee, the Stremelita limao, who is a natural enemy of Tetragonisca angustula. So we wanted to see what happens if we present two very common odors, and we wanted to see if there was any difference if the bee was a forager or if the bee was a guard. So here we have the responses to non-anal. Here we have the stimulus that we present. Air is like a control of a mechanical response. Hexane is a control of the solvent that we use to prepare the different concentration of non-anal. So it's something that you need to do to differentiate the responses of the antenna just because of the odor or is because of the solvent or the air. And what we found here is that the hovering guards present a higher response respect to the nectar foragers. And in between we have the standing guards and the pollen foragers. But if we see what happens with citral, what we see here is that the guards present a higher response respect to the foragers, and again there is a higher difference between the hovering guards and the foragers here. So what we can conclude from here is that foragers and guards can detect both odors, but guards seems to be more responsive to alarm sense, as for example citral. That is something that we expect because if the citral triggers this defensive behavior, we imagine that the guards are more paying attention more to this kind of signals, or they are more sensitive to this. So far we have differences in olfactory and gustatory responses. So the next step was to see if what we found in the ESG or the sucrose response threshold was related with the morphology of the antenna. if there was any difference in terms of types of sensilla or in the number of them. So we use scanning electronic microscopy, and we could identify 6 different types of sensilla. This is what I show you here. And we count the sensilla along all the antenna, and the results show us that there is no difference in the type of number of sensilla present in foragers and guards. Here I show you a table with some of them, but you can see there is no difference. And what we found in all the groups is that the tip of the antenna is this, is what I show you here, is present the highest variety of sensilla compared with the rest of the antenna. Probably this is related with the antennation behavior, because when with the tip of the antenna, the bees touch the flowers or touch another insect or another nest mate. So the conclusions of this part about the experiment related with chemosensory reception is that nectar forager present the lowest score. That means that they have a the highest threshold, sucrose response threshold. That we also find that the difference in the response towards sucrose between pollen and nectar foragers suggests that there is are task related, and also we saw that the other groups, not only the pollen foragers, differ from the nectar foragers. And we saw that both foragers and guards can detect odors, non-aligned citral, but the difference in the antennal responses are task related, since guards seems to be more responsive to alarm sense, the citral. And although we find difference in olfactory and gustatory responses between foragers and guards, we didn't find any difference in terms of the morphology of the antenna. We find the same type of sensilla, and also they were very similar in number. And to finish, I want to show you something related to division of labor, but it's not about the chemosensory stuff. In social insect, the cuticle of each individual is unique, shows a unique profile. It works like a fingerprint. And in particular, cuticular hydrocarbons are involved in recognition of species or nestmates or casts. This is something that is very well studied in apes, mellifera, and many species of ants. And now we have more studies that are showing these kind of things. So our aim was to study the cuticular profile of workers performing different tasks inside and outside the nest. So here we test in hive bees that we took from inside the colony. We opened the hive and we took it from inside. Foragers, in this case, I trained foragers to an artificial feeder, and I took it from them. And as they were foraging for sucrose solution, I can say that they were nectar foragers, and the other 2 groups that we studied were the guards, standing guards or hovering guards. This experiment was done again in the social insect lab at Universidad de Buenos Aires and the chemical ecology lab at the Universidad de la República in Uruguay. What we did was to take bees from these 4 groups of tasks, and we wash them with a solvent, and then we inject that solution in a chromatograph. We analyze the solution using gas chromatography coupled with mass spectrometry. What we found is that we could identify 19 compounds. Here is the list. And also we have some terpenes that maybe they are related with the resins that all these bees carry on their bodies. But we were interested only in the cuticular hydrocarbons, and we found that we have alkanes, linear alkanes, and methyl or branched alkanes, and also some alkenes. When we perform a principal component analysis, what we found was that here we have in green and orange the guard bees, in gray the foragers, and in purple the hivebees. And what we found is that there are like two groups. One is defined by the guards, and we have the second group that you can find the forager and hivebees. Even more, they look like two different groups. But the general conclusion that we can get from here is that the cuticular profile of workers vary according to task group. Guards differ from foragers and in hivebees. If we go deeper and we study the amount of these compounds in the cuticular profiles, we have here the alkanes and the alkenes. We didn't find any difference in terms of alkenes, but there are some difference in the alkanes. Foragers and hivebees present lower amounts of alkanes than the guards. And also we have a difference between hovering and standing guards. So if we go a step forward and we divide the alkanes in branched alkanes or linear alkanes, what we have here is that the hovering guards present more linear alkanes, and respect to the branched alkanes, the difference is between foragers and hivebees and the guards. So again, we can say that the cuticular profile of workers by according to the task group is what we saw before and now, that the guards differ from foragers and in hivebees. The quantitative difference in branched alkanes here maybe is related with the size of the body, could be. And with this, we can say that cuticular hydrocarbons maybe play an important role in nest make and work group identification according to the task that they are performing. So to sum up, what I was trying to show you with this seminar is that division of labor is not only one thing. There are many things that are working in that. And especially in Tetragonisca angustula, what we saw is that in terms of division of labor, there is a temporal polyathism. That means the age can predict the task. And there are some morphological traits related with the labor tasks that the workers are performing, for example, body sizes or some brain morphology. We have differences in the response threshold respect to odors and sugars, and we also see some difference in terms of particular hydrocarbons. Of course, there are many open questions. It's not just, we are just starting with this, and I think that is an amazing subject to study division of labor and try to understand how they can regulate this behavior is amazing. And if you are interested to know more, we recently published this review with Christoph Rutter and Lohan Baladares, and there we show all of the other examples, and also we discuss about all this subject, and also we mark some open questions. And that's all. So thank you all of you for being there. Thanks, of course, Animal Behavior for inviting me. Thank you, the Social Insect Lab at the University of Buenos Aires and CONICET. Please if you are interested in work with or study stingless bees, please contact me. I am on Twitter or X now, and this is my email. We can discuss or just share some thoughts. And also in my lab, we study bumblebees. If you are interested in studying pheromones and bumblebees, please contact Dr. Florencia Palatini, and this is her email. So thank you very much.
Thank you, Sol. So we, at the beginning, we had some technical issues with my audio. Your audio was good the whole time. So I think my mic is good if we can get someone in the chat to confirm, but thank you so much for the seminar. We will move into the question and answer period. So anyone...
Sorry, Amanda, before can I make an announcement?
Of course.
While we wait? Yes.
Of course, yeah, please do.
I want to use this space. I want to share with all of you that this year in Buenos Aires we are going to have the Congress of the Latin American Association of Chemical Ecology. If you don't know Buenos Aires, it's a good opportunity to do it, and if you know it, please come again. And you are on time to use the early bird registration, and you have time to submit your abstract. You don't need to pay and submit the abstract. You can pay now and wait until October to submit the abstract. We have 8 amazing plenary speakers from our region and also from the US. We have 10 symposia that they look very interesting, and of course we are going to have time for posters and oral sessions. So please visit the website or just scan this QR, but that would be super nice to have you here. Thanks, Amanda.
Great, thank you. It's always nice to see lots of societies and conferences happening all over the place. So everyone in the chat is already saying thank you, muchas gracias. Thank you for a beautiful talk, very nice work. So we're getting a lot of feedback already.
Thank you very much.
Just want to make sure that my mic is working. Looks like I will turn down the desktop audio a little bit. It's just sort of troubleshooting in the moment, but it looks like my mic is okay. And okay, great. So just while we're waiting for some questions to come through the chat, I did have a question about the cuticle sort of profile. Is this something that's present from like the juvenile stage, and is it something that changes within an individual as they get older, or is it something that's just sort of stable throughout the life?
No, there is a part that is stable, but of course it changes, because one of the things that change that if the bees are exposed to the environment, they change the profile just to be exposure to the UV or the environment. And if you take bees from inside of the hive, they present these hive odors too, because they are more inside. But yes, it's something that there is a part that is stable, but it can change. Yes.
And it's something that even between, so like for the hive individuals, do sort of all the individuals from one hive have a similar profile, or it's even different sort of within a hive?
No. They have a similar profile. In fact, there are many works in ants that they modify only 1 or 2 cuticular hydrocarbons, and ants from the same nest start fighting because they cannot recognize each other as a nest. But there is, of course, there is like a base, I would say, that cuticular profile that define that you are from one nest. Yes.
That's so interesting. Yeah, I never studied bees, so it's always so interesting to hear about all the different types of research going on.
Yes, now there are people studying cuticular hydrocarbons in wasp or in solitary bees. It's something that is very interesting.
Yeah. And so again, we're just getting a lot of feedback in the chat. So yeah, Francisco, muchas gracias. David says thank you for the fascinating talk. Lohan, thanks, Sol, that was great. Juan, muchas gracias. Kevin, thank you for the beautiful talk. Very nice work. Lovely talk. Thanks for sharing. A lot of feedback. Again, still just waiting for any questions that happen to come through, and we do still have some time. So do you...
Yes, yeah, no problem. I'm happy to be here.
Yeah. Do you have any sort of... I know you did talk about some sort of next steps, but what is sort of some experiments that could build off what you had presented? Do you have anything that you can share that's sort of in the works, or any next steps or big questions in this area?
Yeah, yes. Now I show some data here related with the sucrose response, and I also am studying if there are some difference if you use different sugars like sucrose, glucose, and fructose, because in the nectar this can find these different types of sugars. So I didn't show the data, but I found some differences, and I think that maybe it's not just related with this sense of taste. Maybe there is something related with the receptors. It's deeper, so we are trying to find the response in that sense, in terms of receptors of the antenna or the proboscis, and odor and olfactory and gustatory receptors. Yes, there are many open questions that we are trying to respond, but in terms of course of behavior, we are studying the alarm pheromones or the marks that these bees can leave on the sources. I mean, if they are looking for nectar, they can leave some marks on the flowers, and we want to know how they perceive this mark and how this mark can modify the behavior of other bees, nestmate, non-nestmate, a different species. So we are doing that.
The best part about research is no questions are ever all answered, right? There's always more to look at. So we do have some questions coming in through the chat. So Lohan is saying, hi Sol, I would like to know if you think that the differences in CHC profile among workers could influence somehow the regulation of task allocation.
Maybe yes. It's something that I don't study. I mean, in terms of behavior, I just take the bees. But if we think in the other experiment with apes, for example, yes, there is a correlation with that. So I will need an observation hive just to confirm that. But I think that maybe yes.
Yeah, it'd be interesting to know, is it the chemical profile that produces the status, or the status that produces the chemical profile? It's like, which direction does it go?
Yeah. Exactly. Yes, I think that the beauty of the division of labor is that there is not only one reason. There you have many. As long as you start studying, say, okay, what was first? But it's not only one thing. It's, as you say, is it the task or is the cuticular profile? Yes, we don't know.
Great, awesome, thank you. And David has a question as well. So, how widespread is keeping stingless bees? I recently read that they were the first bees to be kept in South America, but are people keeping them on any scale?
Sorry, again, can you repeat?
So I think he's more so wondering how widespread keeping stingless bees is. So maybe, David, let me know if I'm interpreting it incorrectly, but sort of how common it is, I guess, in research. Like, is stingless bees a huge sort of area of research?
Now, yes, it's going to having capturing more interest from people. Now there are many groups studying stingless bees, yes. And I think, again, there are a lot of species, and we need to know more about them. So there are more and more researchers interested in stingless bees. I think that maybe in the last 10 years, probably, there is an increasement in the groups studying stingless bees.
Yeah. And then I think he'd also mentioned sort of, is it widespread for keeping stingless bees? So do you know anything about like keeping stingless bees outside of research? I guess you can also keep them as a hobbyist. Do you know anything about that?
Yes. Here in Argentina, in the north of Argentina, people have the colonies on the backyards, or they know where the wild nests are, and they go to the forest and take the nest. Yes, there is a very close relationship between people and stingless in the common life. Yes.
That's so interesting. Yeah, I know. It's always that, similar to, some people who study certain organisms, they just end up having them as pets or outside the lab because they just get so involved in them.
Yes, they are pets. Yes, you're right.
Yeah. So we do have another question from Juan. So, hi Sol, I was wondering whether some of the compounds you identified might play a specific role different from recognition, depending on the task that the individual is performing.
Yes. Again, our study was just to describe the particular profile, but we didn't perform any experiment, just manipulating different compounds and to see what was the effect of if they can change or something. It was just describing what we found, and yes, fortunately we got this relation or this difference between task groups. But yes, we didn't manipulate anything.
Mm-hmm. Okay.
It's something that would be nice to do, of course, but yes, you need time.
Yeah, and I guess it depends on the type of bee, because you looked at the foragers, you looked at the hive bees, so the role of these compounds may be different depending on what type of bee it is. So it's, as you said, it's so many complex things kind of piled on top of each other, so trying to identify.
Yes, and I think that, yes, you need to manipulate, I mean, the particular profile in terms of you can add more of one of the compounds, or I don't know, in some way to cover the bee with the particular profile of other bee.
Mm-hmm. Oh, true, yeah.
It's not impossible, but it's something that I didn't do.
Okay, great. Another thing is, at the beginning of the presentation, you had kind of compared the response to like nectar versus pollen versus some other compounds. Do you think there's a difference in response to whether it was sort of a natural compound? Like, say if you took natural pollen or natural nectar versus sort of an artificially created one. I don't know if you compared that, but do you think you would see a difference? Like, do you think they'd be able to tell down to that level?
Yes, I mean, I always work with the natural population. I mean, I don't manipulate the pollen or the pollen resources or the nectar resources. It's true that to test the threshold, I used sucrose. Mm-hmm. And it's what I told you before. I mean, sucrose is not the only sugar in the nectar. So the interesting thing that and is what I am doing now is to try with different sugars and to different mixtures of sugars to be closer to the thing that they find in the nature, Mm-hmm. Yes, but probably yes, there is a difference. There are many things that you can do in the lab, and you can investigate and be closer to what happens in the nature. But yes, it's something that you could do, just manipulate again with real pollen and to produce an artificial one and see what happens. Yes.
Future experiment. Juan does say that it's not an easy task, especially with the diversity of compounds, but it is really exciting, and thank you.
No, thank you.
Yeah. And so, just still waiting for any questions, just trying to think of some other good discussion topics. Well, I guess one of the things, it's up there already, but the Congress that you had talked about, how many years has it been running?
The first one was in 2010.
Oh, wow. Oh, okay. That's really interesting.
Yes.
And is it always in Buenos Aires?
No. It was, the first one was in Uruguay, the once was in Córdoba, Argentina. This time is in Buenos Aires, but the last one I think that was, I don't remember if Colombia or Chile. Yes, also in 2016 was in Brazil.
So it's just all over South America.
Yes.
Okay. So I don't really see any more questions coming through the chat, so I can go through a quick wrap up. But did you have any other sort of final comments or questions you wanted to share?
No, thank you. Thank you, all of you.
Okay, great. Awesome. Thank you. And thank you to everyone who tuned in. Feel free to still post in the chat and I can relay it. But just a few quick announcements in terms of our next seminar will be happening on September 26th. And this will be our last seminar of 2023. Coming up in November as well, we have our annual online conference, which is taking place on November 16th and 17th. The registration to attend, as well as signing up to give a talk, is completely free. Registration is open until the day before the conference, so you can register up until November 15th. And abstract submissions are currently open and will remain open until September 11th. So if you need any more information on either the seminars or the upcoming conference, you can find more information on our website, or you can subscribe to our newsletter, which we send out on a monthly basis. And so that is a wrap on our second last seminar of 2023. Sol, again, thank you so much for the amazing talk. And thank you to our viewers for participating in the chat and watching our seminars. We will see you in the next one. Thank you.